thank you all so much for joining. I would appreciate if everyone could go on mute. And for those of you that would like a language translation, Tyler is on availability here on the chat side of things with directions of how to use Wordly. And I will move forward and let's see if I can move my slide. Okay. So as mentioned, Tyler, thank you so much for your help. Everyone hopefully will be muted automatically. And if you could, type in the chat your name and where you're from and what organization you're affiliated with. If there's any troubles with the connection, let me ask you to turn your camera off. And if you'd like to ask a question, please type the question in the chat. I know there were many questions. We won't be able to get to everybody in the time frame we have, but we will be eager to make sure we capture them and try to follow up with you if you give us contact information to come back to you. This meeting will be recorded. It will be available next week's on YouTube. And I want to make sure everyone understands. Here is our agenda. You guys all may have seen it. I think the agenda for the most part is intact. But one person I wanted to open up and thank everyone from Robert and Julie, our founders, and on behalf of Luca, Frederick, our POLG board, our scientific advisory board, we wanted to welcome you all. And I wanted to introduce Catherine Vassallo. Catherine, I don't know if you want to say hi and just tell everyone who you are and why you're so important to the POLG Foundation.
I don't know about that, but I'm very pleased to meet all of you. I just joined the POLG Foundation at the beginning of June. I have the honor and privilege of being with this foundation. I have a background in finance, so this is a very new world for me, but I had the honor of also being at the Euromic conference, where I was definitely thrown in at the deep end and had 6 days of biology lessons. So I feel it was a great way to start. I hope that I'll be able to help the POLG Foundation, and I look forward to meeting a lot of you. I know I've met quite a lot of you already who were at the conference, but thank you, Brian, for the invitation, and thank you for having me join the board and everything. Thank you.
That's good. Still admitting people, so apologize if I look a little distracted.
Do you want me to reclaim host and I can do that?
No, I think I'm okay. Just as soon as we get into some people speaking, I think I'll be able to pay closer attention to it. But let's start off with this meeting. I think it's called a summit, as some of you may have heard on our first summit, that we've got a mountain to climb, a big monster mountain. There will be a lot of surprises, a lot of challenges, but I truly believe the only way we're going to get there is us working together. And we need everyone together on this one, because in rare disease space and with fragmentation and with so many important causes out there in the world, rare disease is only one of them. And then when you throw in the mix of one rare disease, one form of mitochondria, to me, I think this will really hopefully be an example of a community really working well together, the scientists, the doctors, the patient groups, the families, and we just need everyone because this is not going to be easy where we're headed. But thank you for being on this journey, and we are here for you. And I'd like to first start off with the Pioneer Study, which I hope you've all heard of. It's our natural history study, international collection of countries. The purpose of this is to put together data of rare disease experience of POLG individuals around the world so that we can improve medical care guidance, that we can help inform clinical trials to do efficient trial design, like what endpoints, how long should a study be, and then ultimately could some of our exciting sponsors that you might hear from today consider actually leveraging the data and also considering putting their clinical study arms on top of this footprint. I'd like to invite Danielle Black to say a few words from the MitoShare UMDF effort. Danielle, you might have to ask to be unmuted, and I hope I can
Can
do
you hear
this.
me?
Yeah, please go for it.
Okay. Hi, everyone. As Brian said, I'm Danielle Black. I am the Clinical Research Coordinator with UMDF. I will be overseeing the Pioneer study here in the United States. And we are looking forward to participating in this study and bringing this to the POLG community. So a little bit about where we're at in the United States. So we will have 6 sites in person. They will be Akron Children's of Colorado, Columbia University, UT Health, Baylor, and UCSD, San Diego in California. So what the study is looking like is a prospective multi-center natural history study. And we are inviting individuals with a genetically confirmed POLG associated disorder to participate. Participants can be anywhere from age 0 to 75. There are in-person cohorts available, as long as also including an online platform that we will provide through our MitoShare patient registry. So I think the most important thing I wanted to communicate today is, in order to participate with us, you will have to join in our MitoShare patient registry. And that's the first step. So we are expecting to have some sites active in July and hopefully enroll our first patient. So enrolling in our patient registry is not that a study doesn't need to be active in order to enroll in our registry currently. So if you want to go ahead and do that, that way you'll be all set and we can confirm your genetic testing in order to determine if you're eligible. I'll provide the links to our registry in the chat. So if you are not already registered, you can go there and walk through the steps. I'll also provide our contact information to email. Our email is registry@umdf.org, and we can get you set up there. So we are looking forward to providing this. And then if anybody has any questions for me directly, please feel free to reach out to that email, and myself or a member of our team will get back to you.
Fantastic. Thank you, Danielle. And then from the European side, I would love to invite Marita, if she's on the call, if she can identify herself. Please, there you are. Yes, perfectly.
And you can hear me as well. So nice to be here. Thank you for inviting me. So I'm part of the team in Europe, basically. We have started recruiting patients in Norway, the participants for this study. And we are also including in other European countries, such as Spain, Denmark, and Sweden, Finland. So it will come more countries on the way here in Europe, and also we have one site in the US that will start recruiting patients.
Fantastic.
Do you want me to say more, Brian, or was that it?
Oh, that's good. I mean, that's good. I just, for people out there in the POLG world who say no one's heard of my condition, we're studying it. And we're going to learn so much by combining this collective data from around the world. And we're going to use that knowledge, that new knowledge, in all the possible ways, including our next topic, which is the POLG Externally Led Patient-Focused Drug Development Workshop. We're going to teach the FDA about POLG. I don't know if you've ever given that thought, but the FDA doesn't know about POLG unless we do a workshop like this. And it's going to be amazing. So if I can invite Marina, Kyra, Stephanie, Mindy, if they're online, if they want to mute themselves. I think, Marina, you'll be giving most of the overview for today. Please, Marina.
Thank you so much, Brian. And I didn't know if you received slides to share, or I should go ahead and just present.
You can go ahead and share your slides. Just you might have to ask for permission. I'll stop my sharing and you can do yours. I think I have to just give you permission, but please do.
I think I have issues with because I'm between flights. Actually.
Oh, okay. Give me a second. Let me see if I can pull down your slides.
So sorry for this, but good morning, good evening, good afternoon, depending where you are, and welcome. And on behalf of organizing committee and of this POLG virtual summit, I would like to thank everyone for joining us today to hear more about this externally led patient-focused drug development meeting dedicated to POLG disease. My name is Marina Kolasha. I have a privilege of working in rare disease development and regulatory strategy for 20 plus years. And today I'm here to represent something much more important than regulatory perspective. I'm here to represent patient voice and families living with POLG disease. Externally led PFDD is about understanding what truly matters, to live with the condition and ensuring that those experiences become part of the future drug development and regulatory decision making. Because after the POLG meeting with FDA, we will generate a report that will be part of every drug, every device, every investigational asset that will be submitted to the FDA or EMA as part of as addressing the POLG disease. Probably many of you will have a question, what is this patient-focused drug development meeting itself, right? FDA launched, I'm senior advisor to FDA, and FDA launched the patient-focused drug development initiative in 2012. They were looking to systemically collect patient perspectives on diseases and treatments to incorporate those experiences into medical product development and benefit risk assessments. And as the program evolved, FDA recognized that there are many more diseases than they could address directly, and invited patient organizations to organize so-called externally led PFDD meetings using the same framework, though. Since then, more than 100 externally led meetings have been brought, and they brought together patients, caregivers, clinicians, researchers, regulators, and industry to discuss what's mattered the most for people living with rare and seriously debilitating, life-threatening diseases. And today, POLG disease joins this growing movement. So we will have meeting on the 17th of November, and we are in monthly preparation calls with the FDA to track the progress and our readiness to do the meeting. I will not repeat, Brian. I think we have issues with the slides, but that's okay. We can share them.
Yeah, I think you're doing terrific. I think just keep going. You're doing amazing. And if you can forgive me, I think we're going to forego your slides because you are doing a great job.
Thank you. Appreciate it. I will not repeat that POLG disease is rare and progressive and multisystemic and mitochondrial disorder affecting children and adults, right? And that there are currently no approved therapies specifically targeting the disease. And that was one of the reasons why we initiated this meeting with the FDA. And patient experience, you know more than me that the patient experience seizures and muscle weakness and gastrointestinal dysfunctions and liver disease and movement disorders and neuropathy and fatigue and cognitive impairment, like all the progressive loss of their independence. And despite all these profound challenges, I think many aspects of the patient experience are not adequately captured in the clinical trial. And so that's exactly why PFDD meeting exists. These meetings create an opportunity for regulators, researchers, and developers to hear directly from patients living with the disease, not simply understand the lab result, but understand the daily life and then extrapolate it to the patient-centric endpoints and outcomes for the development. And I think when we were asked by the FDA what are the three messages, when we initially asked for the meeting, we were asked what are the three messages about the POLG, and Kyra and Stephanie, we all shared that first, POLG disease is diagnosed too late. Families often spend months and years searching for answers. By this time, when diagnosis is confirmed, there is irreversible neurological damage that has already occurred. And patients constantly tell us that earlier diagnosis could mean earlier supportive care, earlier clinical trial participation, and ultimately earlier access to the future therapies, that every month is matter. So that was our message number one. And we are welcoming your input and your support, to polish these messages, because this all will be captured in the report. The second is that traditional drug development was not designed for diseases like POLG. Clinical trials were developed for common diseases and large population and validated endpoints. POLG is different, and patient populations are small, and disease progression is very heterogeneous, and children and adults, they are affected differently. That's why we will have two different parts of the meeting focused specifically on children and adults. And many patients cannot participate in lengthy placebo-controlled clinical trials. It's unethical and not feasible. So that's why community was asking a very important question, how we can generate scientifically rigorous evidence while ensuring that patients are not left waiting for years as disease continues to progress. So that was our question number 2, and this will be part of the agenda for the meeting. And the message number 3 was that patient cannot wait for perfect evidence. Like, we need to give up this idea. Patients and families understand the uncertainty. They live with uncertainty every day. They are not asking regulators to lower the standards or approve, for example, risky drug. No, they're asking regulators to recognize that they want to maintain the ability to walk, to communicate, to swallow, to attend school and work and live independently. And that's what these are the meaningful treatment outcomes for them. For POLG disease, stabilization is success already, and slowing progression is success. Maintaining quality of life is success. So this would be part of the discussion with FDA on the 17th of November. So coming more to organizational part, who can participate? One of the greatest strengths of externally led PFDD meetings is that they are not like FDA-led meetings. They bring together entire community. Participants include patients, caregivers, family members, patient advocates, clinicians, researchers, regulatory agencies, medical product developers, patient and caregivers. So patient and caregivers are the main speakers. So they are the one who will speak, who will present, who will pre-record the video, who will participate in the exit polls or survey interviews. But everyone else is here primarily to listen and learn from their lived experience and also provide feedback. And this is what makes these meetings unique. And during the meeting, we will hear from individuals living with POLG and caregivers about their disease experience. They will share the symptoms that matter the most, daily burden of the disease, challenges of diagnosis, their experience with current medical care, their expectations from the future medical care, future drugs, barriers to clinical trial participation, their hopes for future therapy. And participants will also contribute through live discussion. So Kyra will ask you to step in, but they will contribute through live discussion, polling questions, written comments. Like we need to ensure that there are different perspectives are captured, and it's public meeting, so as wide as possible. The goal is not to discuss one specific therapy or some drug in development. The goal is to understand the disease experience and to identify what meaningful treatment benefits look like from patient perspective.
Thank you so much, Marina. I'll jump in just for a second, if it's okay.
Sure.
And
Please,
just what an
Karen.
honor it is for us to host this meeting in partnership with the POLG Foundation and to have Marina and her incredible expertise to help us serve as our moderator and guide us through this process. But I just put in the chat, as Marina said, your input is critical to this meeting. So I just put in the chat a link to our website page where you can actually go in and share your comments about your journey with POLG. And this information will also be shared with the FDA and included in the Voice of the Patient report. And then in the coming weeks, we're also going to be launching a community survey. And we would welcome everyone to participate, share your journey, answer questions, because that will help guide some of the content that we provide with the FDA, knowing the pain points we need to focus on. And then those survey results will also be shared with the FDA and included in the Voice of the Patient report. So there's lots of opportunities for you to participate, and we'll continue to share those with the community.
And as Kyra mentioned, the communication with FDA doesn't stop with the meeting. When the meeting ends, no communication anymore. No, it's not the case. There will be a report. The report will summarize everything what we've heard and all the information that we received to date. And this report will be submitted to the FDA. It will be publicly available on MitoAction POLG website and also reshared by FDA on their website. Those, we also propose keeping public comments period open for, I think, 1 month after the PFDD meeting, so that the patients and families who could not participate live, they still have an opportunity to contribute their experience and perspective. So every voice matters. We need to be sure that we captured wide, all the perspectives. And why it matters, because I'm working in rare and orphan for 23 years, and I've touched and I'm responsible for 299 drug approval, both sides of Atlantics. And I know that my history shown that patient perspective changed drug development. These perspectives influence endpoint selection. The perspectives improve clinical trial assessment. They guide benefit risk evaluation and discussion with regulators. They help researchers to understand what truly matters to the patient. They remind us that behind every clinical trial is a person, is a family, and community waiting for hope. And if I want to finish, and I'm sorry, I think I took longer than expected, but the POLG disease affects every aspect of daily life, and we have this opportunity to ensure that patient experience becomes an essential part of regulatory science. The success of this meeting will not be measured by minutes or a report or who was presenting. It will be measured that whatever every patient feels, that their voice, if every patient will feel that their voice is captured, is heard, and their voice will help to shape the future therapies, clinical trials, and regulatory decisions. And I think together we can build this opportunity where treatments are developed not only for patients, but with patients. And on behalf of MitoAction and entire POLG community, I would like to thank you for listening. I'm sorry for taking longer. And thank you for your commitment to accelerate meaningful therapies for everyone living with POLG. I look forward to continue this dialogue. Thank you.
Thank you, Marina. If I could also ask Stephanie Harry to say hello. That's an important voice we absolutely don't want to ignore. Stephanie, would you please say hi?
Hi, my name is Stephanie Harry. I'm the patient support coordinator at MitoAction, and just appreciate you guys. Kind of to tag what Marina said at the last part, to me, the most beautiful part of the PFDD meeting is the fact that you have opportunity to share your stories, and our stories change things, and there's power and wisdom in our stories. And so thank you so much for the vulnerability that each of you have had, whether you've attended our support calls or you've been in communication with me. Our stories matter and can change things. And so I'm super excited to be able to work with you guys and sharing your stories. And if you're curious or interested in the PFDD, or if some of this is new to you, just reach out to me. I put my email in the chat, and there's lots of different ways to get involved. If you're like, I don't want to be on screen, that's too much. There's so much more ways that you can help that are just as equally as important. And we need everybody's help. It's a community effort. So thanks so much, Brian.
Thank you, Stephanie. I'm just going to underscore this with one last comment about the patient-focused drug development workshop. This will be the most important day in the history of the world for POLG and medicine's development. I'm going to just pause with that thought. I don't want any of you to think that this is the POLG Foundation or MitoAction. This is your workshop to teach the FDA. They don't want to hear from the doctors and scientists and nerds like me. They want to hear from families. They want to hear from those of you that live with POLG. It's a big day, and we've got to capitalize on that day. So please, in any way you can participate, we want you. And I see we have various patient organizations, UMDF, IMP, anywhere around the world. I want everyone to know that this is for the POLG community. And if you're supportive and there's things we can put together of the broader community of patient advocacy for POLG in the Mito world, this is an amazing opportunity for us to take full advantage of. With that, I'd like to introduce Carlo Viscomi from Italy, who is one of our POLG champions. And I say that from the bottom of my heart because I've watched Carlo in action for many years now, and some of the work he has done is really paving the way of how we can leverage the best of science to become medicine. We can't do it without scientists like Carlo, because Carlo is so smart, he could study anything, but he's studying POLG. And so, Carlo, please, if you can unmute yourself, and I think if you have slides to share, I will absolutely give you permission to share if we can do this.
Thank you, Brian.
Please, Carlo.
Thank you, Brian, and hi, everyone. Sorry, I'm trying to understand how to share my presentation. Let me see. So while I'm trying to launch my slides, I wanted just to tell all of you that for me, as a scientist, having the
Can see your
chance to
slides,
interact,
Carlo.
yes,
Perfect.
Having the possibility to interact with the patients and their associations is empowering, and it's essential for me to keep the target and the goal of my research, which is not just describing interesting basic aspects related to mitochondria or cell biology, but it's about being able to help the patients by trying to identify new therapies. So I think that you are all more or less familiar with this. Our cells contain organelles which are called mitochondria. And what mitochondria do is to convert the energy that we get from the food we eat into a form which is immediately available to our cells. And mitochondria are unique in our cells because they contain one specific molecule, which is represented here, and it's called mitochondrial DNA. And that's essential for the functioning of mitochondria. And when something happens into mitochondria and in mitochondrial DNA, mitochondria do not work properly, and then disease can arise. And these diseases can affect different organs, including the brain, the skeletal muscle, and the heart, which are the most energy-consuming organs in our body, but not only, because also the liver and the kidneys can be affected. And this is what happens when there are mutations in POLG. POLG is a gene encoding for or specifying the sequence of the protein which copies mitochondrial DNA. And so it's essential for the maintenance of mitochondrial DNA. Mutations in POLG are associated with a wide range of different conditions which are represented in, which are represented in this slide. Over 300 mutations have been described in POLG, and they can determine very diverse diseases, some of which are very early onset, and these are usually the most severe ones, while others arise at different stages in life, as it has already been mentioned here. In any case, the common thread of all these conditions is that they are characterized by the accumulation of or alterations in mitochondrial DNA. In some cases, they are associated with a reduced copy number of mitochondrial DNA. We have massive amounts of mitochondrial DNA in our cells, and the reduction in this amount gives very severe syndromes. A bit less severe are mutations associated with so-called multiple deletions, i.e., smaller, shorter forms of mitochondrial DNA. Among all these mutations, some are particularly frequent, and they are represented in this slide. I don't want to go into all the details, but this slide only helps me to point out that some mutations are more frequent than others. Why is it so difficult to investigate POLG-related disorders? Because of this extreme complexity, number of mutations, different symptoms, and obviously we cannot just take samples from the patients. we need, in order to be able to investigate how these mutations are associated with a certain range of symptoms, we need models. And models can be cells, can be organisms, like in the case, in my case, mostly mice, but also in the case of other colleagues I saw in the audience. And they are essential to understand the mechanisms causing the disease, but also to test new therapies. And this is essential to then go back to the patients and test whether these therapies that we can test in the models can be effective in the patients as well. In this frame, we reproduced the most frequent mutations in POLG in mice. And this table summarizes the fact that we have generated a large number of mice and started a big project to try to reproduce the genetic and hopefully clinical complexity of POLG-related diseases in mice. So we generated these animals, and we are characterizing them. I don't have the time to go into all the details, but I'm going to show you just one specific feature we detected in our models, which is represented here. This is a skeletal muscle in a normal mouse. It can well be a human skeletal muscle. And these dark spots that you can see here are normal mitochondria. Sorry, this is. In the lower panel, you can see the abnormal mitochondria present in one of our mouse models. And you can see that they are massive, they are gigantic, and they have these white spaces which are clearly not present in wild-type mitochondria. We identified one drug and we tested it in our models, which is able to interfere with the accumulation of these gigantic mitochondria. And if you can see here, this is a mutant, the skeletal muscle of a mutant mouse after the treatment with this compound. And you can clearly see that mitochondria are much smaller. They are still not completely normal because these white spaces are not normal in mitochondria. However, compared to the untreated mice, we can see that we can also identify several normal mitochondria, which is the panel on your right side down here. And this is one of the reasons why we focused so much on mice, because we can test new therapies for POLG-related disorders. This is not the only approach that can be tested. The advantage of this treatment is that in principle, it can be applied to several different POLG-related disorders independently of the underlying genetic defect. There are other approaches which are much more focused on the single mutation. And this is the case of the so-called base editing. Basically, it's a way of correcting the mutation. I don't have the time to go into the technical aspects of this, but there are basically two reagents that we are currently testing in our cellular and mouse models of POLG-related diseases to correct the mutations. And these two tools, which are called technically CBE and ABE, meaning that they are specific for certain mutations, can cover large part of the mutations in POLG and basically in any other gene. So basically, just to make an example, you see here in this stretch of DNA, you have GC, and this CBE reagent can convert it into TA. So if TA is the normal base pair in the DNA in POLG, and GC is the mutant residue, it can be corrected. This is not going to happen next month, probably not even next year, but that's where research is moving, at least part of the research. This is not the only approach that can be taken. I'm sure that my colleagues will show different approaches in their presentations, but this is where my lab is currently moving. And I think I'm done with my presentation. I just want to thank Brian and the POLG Foundation for organizing this meeting, and my group because they are actually doing the job. And I thank you for this attention, and I'm happy to take any questions.
Thank you, Carlo. If you have questions, go ahead and just type them in and we will send them on to Carlo. I'd like to invite the next speaker, who's also quite a champion for POLG, and he's got some research that is really on the cutting edge. D, if you can go ahead and get your slides ready, I'm happy to give you permission to share.
Certainly, Brian.
See if you can try to share.
Yep. Can you see my slides?
Oh yeah, you go. You're ready. There you go. Take it away, please.
Metodiev. I'm a researcher with the French Health and Medical Research and also the Imagine Institute in Paris, also known as the Inferno. Currently, so Carlo actually introduced the genetics and touched on the pathomechanism of POLG disease very nicely, which gives me the opportunity to forego any lengthy introductions and to practically get right into the things. So I would like to specify that what we are doing is in fact a gene replacement therapy for POLG disease. And just to remind you, the idea here is that when we have a cell carrying a mutated POLG gene, and this gene produces a dysfunctional, causes a mitochondrial dysfunction, we want to actually supplement associated virus or other delivery vehicle to provide a functional copy of the POLG, which then will produce a functional POLG protein and restore mitochondrial function. So when we were starting this project, right at the outset, we had to face two challenges. First, the first challenge was that at that time, we didn't really have any good mouse models. And by good models, I mean mice with robust clinical presentation that would help us define clear endpoints. We didn't have this vast library that Carlo has created in the last years. And the second obstacle that we faced is the large size of the POLG cDNA, which far exceeds the cargo capacity of classical adeno-associated viruses, which are typical delivery vehicles for gene therapies. So who created for us a conditionally targeted mouse model. And the advantage of this conditionally targeted mouse model is that it enables us to actually choose where we want to inactivate POLG genes in different tissues and organs. But this approach, of course, leads to complete loss of the POLG protein. And this is different than humans, of course, but for our project, it has the advantage that because of the essential role of the POLG protein, the knockout mice invariably develop a very strong phenotype. And actually, nowadays, these mice are part of the POLG Foundation toolkit, and those that are interested, scientists, researchers, and clinicians can access those mice to checks. So we use these conditionally targeted mice to inactivate POLG in what we call parvalbumin positive cell types. The important part for the question here is that these include neurons, which are responsible for motor coordination and also neurons involved in seizures and other clinical presentations linked to POLG disease. Now, what we observed immediately is that actually POLG mice develop a progressively worsening motor coordination deficit. And this is a little video here just so you can appreciate the phenotype. One moment, let me start it. There we go. So to the left, you can see a normal mouse, a control mouse, which exhibits a normal exploratory behavior. And to the left, you can actually observe the consequences of inactivating POLG. One moment, I lost my slide somewhere. There we go, which has lost POLG in the. Can you see the animation right now?
Yeah, I could see it when you had it running the first time. It looks like maybe it's hung up now. I don't know.
Let me share again. Let me share again.
Okay, and maybe if you want to turn off your camera, if bandwidth.
Yeah.
Maybe if it helps a little, I don't know.
Let's do that. Oh, I can't find the correct. Oh, let's try again. Do you see the video?
Yep.
All right. So that's basically how we have a very strong. I should point out that this is actually a very advanced disease, and we don't typically keep the mice to that stage because of European animal welfare laws. These mice also exhibited a shortened lifespan, and you can see here that for the group, the median lifespan was actually 22 weeks. So now we have a very nice model with two therapeutic endpoints. We have lifespan or death at 22 weeks and progression. About the vector, I cannot go into any details regarding the vector, but we tinkered around with the vector design and we were finally able to produce an optimized vector, which enabled us to accommodate the full-length POLG cDNA and to use a approach for our work. We then proceeded to generate AAV9 pseudotype viral particles. These are viral particles that has the potential to target different organs and tissues in the body. We injected these particles into neonates. So we chose newly born pups because soon after birth and for a few days and weeks later, we are actually able to target the brain. And we then proceeded to monitor the pups over time. After a certain age, we started a type of analysis called rotarot. This analysis basically is based on the fact that mice with motor deficits cannot hold onto a rotating rod. And so we control mice usually have no problem to stay on the rotating wheel, but knockout mice inevitably lose this ability. And in fact, by 19 weeks of age, all of our untreated knockout mice were not able to were consistently failed the rotarot test. In contrast, treated mice up until 19 to 23 weeks were indistinguishable comparable to controls, and their rotarot performance then started to decline a little bit, but for the duration of the experiment, it never reached zero. And in fact, at 35 weeks of age, we decided to stop the experiment because we have achieved sufficiently statistically significant extension of the lifespan. All of the mice except one, all of treated knockout mice except one survived 35 weeks of age. Thus, this little video here. So even though the rotarot performance of treated knockout mice declined a little bit with age, they really look indistinguishable from controls. And to the left, we have here one control mouse and one untreated knockout mouse. This mouse is younger than the mouse that I previously showed you, but you can, I hope you can see this specific wobbly walk of the mouse. Clearly, there is something wrong with the mouse. And to the right, you can four treated POLG1 knockout mice. let's play both together. You can see that these mice look quite okay, even if they have a little bit decreased overall performance. Our work so far showed that a single high-dose injection in newly born mice was able to preserve respiratory chain activity in POLG deficient neurons. I did not show you this data, but we had extensive histological analysis of mouse brains and were able to actually observe this. This treatment also delayed the onset of motor coordination deficit lifespan of treated knockout mice, at least until 35 weeks of age. And so far, the data look very promising for us, but I have to say that our work is not yet done. We still need to know what is the maximum extension of lifespan that is conferred by this treatment. Are lower doses actually sufficient to protect against the development of the disease? And what happens if we treat our mice before or shortly after the onset of disease? Can we actually stop the progression of disease, which I think is also a very important point. And finally, I would like to acknowledge my team members and as well as our collaborators in the phenotyping and AAV platform at the Imaging Institute. And of course, the generous support of the POLG Foundation, because without you guys, this project would never have taken off and have never have worked. And also would like to acknowledge my other. Thank you. And I'm waiting for your questions.
Incredible, amazing, Matodi. I don't know what you guys think about that, but you just saw something that's astounding. It hasn't even been published yet. You're getting a preview of something that still the scientific community doesn't even know about yet. But Matodi, thank you so much for sharing your amazing work. And we want to see where this can go as fast as possible to the clinic safely. And when you say funding from the POLG Foundation, I want to say the funding coming from many of you who support us. And this is what the places we want to put money where we think it's got a path to get to the clinic and to be hopefully a treatment. And I hate to use the word, but could this be a cure? I see some questions popping in. I'll be sure to send those on to you, Matodi, but I'm going to keep moving because we got some other amazing talks to do. So thank you so much to Carlo and to Matodi for on the basic science, early preclinical side of research. It's amazing, right? But now let's shift into some of the human form of research, of which are also extremely important to the clinic. And next up, I'd like to invite Thiloka Ratnaki from Bristol. She's doing some pretty clever work in leveraging some AI. So Thiloka, if you want to open up and try to share your screen, your deck, please.
Thanks so much for having me. I'm just going to try and get the presentation up. It's such a great meeting as well. Second, there we go. So share. Can you see
your screen? Yes, we can.
Perfect. Can you see it in the right
Perfect
mode? Perfect. Good. Okay. So I'm Thiloka. I'm a pediatric neurology registrar and early career researcher, currently in an honorary capacity with the University of Bristol and Cambridge. And these are some of my recent funders. And I wanted to talk to you about what I've been doing to think about those clinical features of POLG and how we can kind of better use data to inform diagnosis. And that's so important if we are already at the stage of trying to develop treatments as well. So to get to that, I want to quickly go through how we diagnose mitochondrial diseases in the UK, but it's very applicable to a lot of settings. I'll talk about the phenotype challenge, and by phenotype, I mean, the clinical feature or set of clinical features, symptoms and signs which go along with a condition. And then what we've been doing to improve the workflow in 100,000 Genomes Project, which is a large rare disease study in the UK. and then how we've been going about kind of harnessing the data to think about machine learning to improve efficiency, let's say. How we diagnosed Mito in the UK. So this is a paper we published in 2020 when whole genome was sort of being set out as a first-line diagnostic test. We were suggesting, look, if you're suspecting a mitochondrial disorder, you should be moving fairly fast to do the whole genome sequencing. And in some cases, you would do muscle biopsy because that is still a gold standard for some mitochondrial diseases. But working away from, step-by-step diagnostics, which extends that journey. But this is where you come to the phenotype challenge, because to think about the suspected mitochondrial disease, you had to first kind of recognize a set of clinical features. And this challenge is not exclusive to POLG. Sadly, it's, felt by the rare disease community. When you have a rare condition, it's very much almost potluck whether the clinician has thought about it. And then, as lovely as we all are, clinicians also use variable descriptions of the same features, and that means it's very difficult to kind of quantify the characteristics of a condition. The other thing is, as we know, some parts of the condition can mimic common conditions, and we try and exclude the common things first. Again, this all adds to your diagnostic sort of process. So to harmonize the language that's used between clinicians and researchers, a database called the Human Phenotype Ontology has been developed by the Robinson Group in 2008 and has been evolving since then. And what a HPO is a more structured term to capture what different people mean in this example by short stature. And each term in this database is connected to either broader terms that they belong to or more specific terms that come from them, so the kind of the leaves. And that means from a research perspective, you can start quantifying what the clinical characteristics of a condition is. And what I've done is painstakingly gone through a lot of literature in mitochondrial diseases to capture the individual level characteristics of mitochondrial diseases, and POLG is one of the the biggest data set in the nuclear encoded mitochondrial diseases. And then when we have the individual level phenotypes captured with the genotype, which means gene and variant, I can then do bird's eye views of the data and say, look, this is how different POLG can look across the different age groups. I could also go and do a deeper dive into the data set, and this is me going, these are the top 20 terms in this MitoFen data set for POLG, and look how differently they're spread out between the different ages of onset. And yes, there's overlap, but also there's some interesting and important defining features as well. And that's all important because that means we have a rich data set to then think about how we can add that into our diagnostic pipeline. And if I know that there's a lot of rich data here, I can then start manipulating it statistically speaking to see whether there's a special characteristic about POLG that defines it across mitochondrial diseases. And that's basically what I've done. Within MitoFen, we've managed to show that, that there's a phenotype similarity score, which is specific within POLG enough that you can fairly confidently pick out POLG against other mitochondrial diseases. And we've applied that sort of ground truth to the 100,000 Genomes Project, where we have, diagnosed and undiagnosed patients with rare diseases. We've looked for everybody who has a POLG variant and HPO terms available, applied the phenotype similarity score using MitoFen, and we've picked out additional individuals who don't have a diagnosis yet, and we're working them up to make sure that we get it right as well. So kind of moving a bit forward, so we know we have that rich data set helps, but how can we make that process efficient? Because as soon as I publish MitoFen, it's already out of date because there's so many new papers coming out all the time. So we're now with Bristol using the MitoFen dataset to better adapt machine learning approaches to make this whole process a lot more efficient. And you'd be surprised to know that, large language models like ChatGPT don't map HPO terms from your free text accurately at all. But this kind of approach that we've been developing in Bristol has sort of an over 90% accuracy, which is something that we haven't published yet, and we are testing now as well. So future directions, I want to generate diagnoses for our undiagnosed individuals, test this machine learning approach at scale, and then I want to apply it to the electronic health records because that's got a lot of potential, in the natural history studies that have been talked about to try and capture the kind of data that we're not biasing by suggesting the types of outcomes. And then I also want to know, what am I missing by only focusing on the published literature? Where is it that these large language models could help us with what patients and families experience? So that's the kind of strategies that are upcoming, and hope you're interested, and I'll keep you all updated. These are some of the acknowledgements as well, and thank you.
Thank you, Thiloka. We just have to do better for our POLG families. And getting to a diagnosis is some of the hardest parts of it. So we appreciate you doing the work you're doing and look forward to seeing further evolution of it. I appreciate anyone who has questions for Thiloka. Please put them in the chat and she can try to tackle them. I do know she's on call right now, and so if she doesn't get to today, I understand why. But thank you so much, Thiloka.
Thank you.
I did want to, you bet. I wanted to ask if Bill Copeland could just say hello if he's still on screen. A lot of times we wonder, how do we know what's a mutation or what's not a mutation? Bill, are you on the screen still? I think you are. Bill just recently joined our scientific advisory board of the POLG Foundation. And
hey,
Bill,
Brian,
if you want to say
hear
a
me?
few words, yeah, please introduce yourself,
great.
say hi, and I would love to hear what you thought of what Thiloka is doing to help us get our diagnosis more accurately.
Definitely. It's been needed badly, and I'm really happy to see the all coming together to form a more concise sort of collection of symptoms to help the diagnosis. But yeah, it's
wonderful.
Excellent. Yeah.
Excellent. Well, thank you, Bill. We're so excited to have you on our scientific board. Next up, we are going to have an amazing talk for those of you that said, what's the connection of POLG and epilepsy? It's a murky answer. It's very difficult to explain that. And it's something we all burn with. Like, help us understand this, please. So there's some amazing research going up in Newcastle, UK, and Bobby McFarlane's team. If I can invite Laura Smith and Syed Ahmed to jump in. They've been diving into this question and would welcome them sharing their insights and where they are with their research.
Thanks, Brian. Can you hear us okay?
Perfect.
And you can see our slides?
Yes.
Okay, perfect. Okay. Hi, everyone. Firstly, thank you so much to the POLG Foundation for inviting us, myself and Saeed, to talk about our research, which involves investigating what could be causing epilepsy in POLG-related mitochondrial disease. So we know that epilepsy is a very common neurological symptom of POLG-related disease, which often can involve very frequent drug-resistant seizures, which are very difficult to control. We also know that these seizures can be associated with stroke-like episodes, which are essentially a new onset or worsening of neurological symptoms. This epilepsy also often involves the occipital cortex in the back of the brain for reasons we do not yet fully understand. And unfortunately, because this epilepsy can be very resistant to treatments, it's typically associated with progressive neurodegeneration, which is linked to neurological decline. But the goal of our lab is to better understand what's causing this epilepsy, so hopefully we could then identify new ways to better manage seizures for patients. So we are particularly interested in understanding what specific types of brain cells could be driving these seizures. We are also investigating whether inflammation could be playing a role in this epilepsy. We would like to better understand why the occipital cortex is so involved. And in our lab, we're also developing new models to investigate POLG-related epilepsy. So first of all, just a little bit about the brain and what seizures are. So we know that our brains are composed of billions of neurons, and there's more than 100 different types of neurons, which all have very different functions and properties. But together, these neurons generate electrical-like activity through a process known as neurotransmission. And this electrical activity needs to be very regulated in our brains. So there needs to be a balance between neurons which stimulate and generate this electrical activity and neurons whose jobs are to dampen down and slow the brain activity. And we know that this electrical activity is very energy demanding, so it depends a lot on our mitochondria, POLG, and our mitochondrial DNA. Seizures are essentially a sudden, uncontrolled burst of electrical activity, which can occur when there's an imbalance and essentially leading to an excessive buildup of electrical activity. And finally, we know that dysfunctional mitochondria in the brain can disrupt the normal functions of brain cells, but we don't yet fully understand how this directly causes epilepsy. And this is the main focus of our research group at Newcastle. So how can we investigate what specific types of brain cells could be driving seizures in POLG disease? So in order to investigate this, we examine donated brain tissue samples from patients with POLG disease, and we can compare these to age-matched control brain tissues. And using very specific antibodies, we can label specific cell types and proteins on very small samples of brain tissue, and we can study these under the microscope. And this allows us to identify which mitochondrial proteins and which specific types of brain cells could be altered or perhaps absent in POLG patient brain tissue. So to summarize some of the findings from our studies using this approach, we have identified that there seems to be quite a severe loss of key regulatory neurons in POLG patient brain tissue, known as inhibitory interneurons. So in these images here, these brown cells are these key regulatory neurons, and we can see that in POLG patient brain tissue, there is quite a severe loss of these cells. We've also observed changes to the main support cells of the brain, which are known as astrocytes. So in POLG patient brain tissue, we observe a huge accumulation of these support cells, which also appear to have altered functions. And similarly, we've also identified an accumulation of the immune cells of the brain, which are known as microglia. And based on what we know about these three main cell types, we think that there could be a loss of these key regulatory mechanisms in the brain, which prevent electrical activity from being dampened. So this could in turn promote the seizure-like activity. So we're also interested in understanding what else is happening in the brain. So what pathways and processes could be altered? So to investigate this, we have also been examining the donated brain tissue samples, but using a slightly different approach. So for this, we've taken very small samples from two different brain regions. We've homogenized or broken down these samples, and we've analyzed them using a technique known as mass spectrometry. And this essentially allows us to quantify thousands of proteins which are present in this tiny piece of brain tissue sample. And using this, this method, we have recently identified that there seems to be an increased amount of innate immune system proteins in POLG patient tissue. And there's also quite a lot of evidence suggesting that there's inflammation. And this is just a snapshot of some of the data from this study. So this study has suggested that inflammation in the brain, known as neuroinflammation, could be playing a role in POLG-related epilepsy. But we're now actively investigating whether this inflammation could be a cause and or a consequence of seizures. So I mentioned that the occipital cortex, part of the back of the brain responsible for our vision, is quite often involved in this epilepsy, but we don't really understand why. So to better understand this, we've used a similar approach where we have compared brain tissue, control brain tissue, from two different brain regions. So the occipital cortex, which is often involved in this epilepsy, and we've compared it to the frontal cortex, which is typically less involved in POLG disease. And we've used the same approach where we've analysed thousands of proteins present in these brain tissue samples of control tissue. And interestingly, what we observed was that there were significantly more mitochondrial proteins present in the occipital cortex of the control tissue. So more than 100 mitochondrial proteins were more abundant in this brain region. So this suggests that perhaps the occipital cortex has a higher metabolic or energy demands, and this could explain why this brain region seems to be particularly vulnerable to dysfunctional mitochondria, and it could partly explain why the occipital cortex is so involved in this epilepsy. So I'm now going to pass you on to my PhD student, Saeed, who's going to talk to you a little bit about how we can, based on what we have identified in patient brain tissues, how we can use this to model features of the epilepsy.
Thank you very much, Laura, and thank you, Brian, and thank you for anyone joining us today. So as Laura mentioned, the innate immune system signature was quite upregulated in the donated brain tissue that we were studying. So in our lab, we're currently interested in actually studying these immune cells in our brain and how they behave under POLG depletion and the role of POLG disease itself. So how to do that is we could obtain these cells using a small skin biopsy or, for example, blood. And scientists have successfully reprogrammed the skin biopsies and the blood cells to get them back into the stem stage, basically meaning that they could give rise to any cell lineage or any cell type that we require to study in the body. And these cells were generously donated by the POLG Foundation toolbox, and we're currently using these cells to model POLG-related epilepsy and to understand the dysfunction of POLG in these cells and how would that interact with the neurons surrounding by and how would they will all lead to neurodegeneration and hyperexcitability. And also our work currently is using a human microglia cell line, which are called HMCC3. And these cells are a proof of concept of the microglia, which are the brain immune cells, which are responsible for the removal of debris and the pathogens that could invade the brain, and also they are neuroprotective from any infections that could happen in the brain and for the neurons as well. And these cells, we currently using them as a larger scale and obviously a cheaper way to kind of optimize our methods before we go into a physiologically more relevant way of using the inducing pluripotent stem cells that we've obtained from the POLG Foundation. So as a final to that, we would like to thank everyone who generously donated to get this work done, to all the patients and to our lab as well. Thank you very much.
Incredible. Thank you, Laura and Syed. This is such a burning question for our POLG families, our clinicians. I'm really excited about what you have here. And if you guys can help us figure out how to treat it, how to solve it, how to prevent it, amazing. So I hope a theme that everyone's picking up on is leveraging the best of science, the greatest minds is what we need on our team to climb this summit. So Laura and Syed, and please all our best to the wonderful people in Newcastle that are fighting mito disease. We celebrate and we praise you so much. So thank you for your efforts. And as I understand, that's not published work yet either. So you're sharing data with us that no one has seen yet. But this is for the POLG community around the world. And boy, we have people calling in from around the world. So I'm hoping people are appreciating the importance of really top-notch science being focused into POLG. So thank you.
Thank you.
Thank you.
Next up, we have on the theme of best of science and in human research, and how does perhaps viruses and immunity in children with mitochondrial disease have the impact that it does. I'd like to invite Peter McGuire and his team from NIH. I have your slides, Peter. I just need you to
I can try sharing. I'll see if it'll work
Okay.
on my end.
All right, go for it. Peter, please do.
Here.
Thank you.
Okay, let's see.
We can see your screen popping in.
Okay, great. All right. It's a good day when your IT stuff works. Okay, let's see. Okay, so Brian, thank you very much for giving me a chance to talk a little bit about our study and kind of summarize some of our findings. And also, I wanted to mention at the end, we have a very exciting new initiative that's specifically for families or children affected by POLG disease. So I'm the PI for the NIH mini study. I work at the National Institutes of Health in Bethesda, Maryland. The NIH mini study, mini stands for Metabolism, Infection, and Immunity in Inborn Errors of Mitochondrial Metabolism. And this is a study in children of viral infection and their immune system. So why do we study children? Well, one of the reasons is because the immune system is very different in children than adults. During the first 10 years of life, that's kind of a period of challenge where the immune system has to learn to be able to react to various viruses and bacteria and be able to protect an individual from those things. And so this is a longitudinal natural history study that we run at the NIH Clinical Center, that we invite families with a child with mitochondrial disease from all over the country and even all over the world. And they spend a couple of days with us and undergo numerous evaluations. And so this is the team here. This is myself here on the left. And then there's also Eliza Gordon-Lipkin, who is our pediatric neurologist and neurodevelopmental specialist. And then Shannon Krupp, who's our research nurse who handles and coordinates a lot of the visits. So just to start out, I know that we all think about the immune system in terms of infection, but we should, also it's important to point out that the immune system is involved in many other things besides just fighting infection. So it's involved in things like brain and neurodevelopment. It's involved in energy and extracting energy from your food. It's involved in the ability to protect your gut essentially from microorganisms. It's also involved in things like inflammation, of course, which I'm going to talk about. I know it's been a great lead-in terms of inflammation. Infection response, as I mentioned, and also it's involved in other organ systems, especially in recovery and healing. So a lot of other functions beyond just infection. So there is an interaction between, or a two-way relationship between mitochondria and the immune system, where immune cells require a lot of energy to be able to function. They have to proliferate and make more immune cells, and they have to be able to fight off viruses and bacteria and recognize a whole broad range of various infectious organisms. And so that requires a lot of energy. And so dysfunctional mitochondria can affect the immune system. But at the same time, the immune system, when it becomes activated, we heard before about neuroinflammation. There's also generalized inflammation in individuals with mitochondrial disease. And the reason that's problematic is because tissues that are affected by mitochondrial dysfunction do not tolerate inflammation. Inflammation is essentially immune cells secrete these small, I like to call them text messages. They're like cytokines, which immune cells use to communicate with each other. The problem is tissues affected by mitochondrial disease don't like those text messages, and that tissue can become dysfunctional or even more dysfunctional in the setting of mitochondrial disease. So some of the things that we've noticed in a number of the families that we've evaluated, one thing I forgot to mention, the mini study's now been around for over 10 years. We've evaluated hundreds of patients with mitochondrial disease, all different types, including patients with POLG disease. And some of the themes that we see emerging from some of the children that we've evaluated is that, first of all, infections hit harder. They can last longer. The kids can get sicker. It takes a long time to recover. Some symptoms that the individual actually has actually worsen after illness. So for instance, we published a paper a few years ago that with viral illness, and these are respiratory viral illnesses, that seizures can actually get worse in individuals with mitochondrial disease. And actually their seizure frequency can actually increase after a viral infection and remain persistent. And so those symptoms or that exacerbation of symptoms can actually persist even after the infection is cleared. So the reason for this is that the immune system may be out of balance. And so some of the observations that we've made is that inflammation actually can occur in mitochondrial disease even in the absence of infection. So this was a study we published a couple of years ago where we actually noted in a cohort of about 30 patients with mitochondrial disease versus controls, they had markers in their immune cells of inflammation, of ongoing inflammation. And this was in the absence of infection. So this smoldering inflammation at baseline can actually get worse or become overactive during infection. And that actually can produce a lot of adverse effects and increased morbidity in children with mitochondrial disease. On the other side, the inflammation deals mostly with the innate immune system. On the other side, the adaptive immune system, which handles immune memory. So in other words, the immune system learns about viruses and remembers them and tries to fight even harder next time it sees them. Those memory responses can also be weaker or even delayed. So why does this actually matter? Obviously, for patients and for their for their well-being, it can help explain day-to-day health challenges. As I mentioned, viral infections and the inflammation that results with viral infections may influence disease progression. But also, we need to understand the immune system in mitochondrial disease because this is critical for future therapies, whether it's gene therapy or small molecules. The immune system touches every single aspect of mitochondrial disease. So who is the mini study for? So children with mitochondrial disease. And so we want to see everyone, even whether you have had frequent infections or think you have difficulty with the immune system or inflammation, even if things actually seem normal, so to speak, your participation actually matters because it's important to understand individuals who do not seem to have issues with viral infections or immunity, as well as those who do. So we're able to make those comparisons. So some of the evaluations that individuals receive when they come to the NIH Clinical Center include things like an immune evaluation. They also get neurodevelopmental assessments, and a lot of families have found this very helpful because in the outside world, this is testing that you oftentimes have to pay for, and it can be helpful for educational planning and schooling, essentially. Patients also receive a neurology evaluation, nutrition assessment, rehabilitation medicine is also an important part of what we do, and as well as ophthalmology evaluations. So all of these consultations at the NIH Clinical Center are provided at no cost to families. So to help with participation in research, we try to remove a lot of those barriers for research participation. So we provide resources and support to every family so that they can participate no matter where they live. So we have families who come from the United States, we have families who come internationally, and we provide things like travel support and lodging as well as meals, and try to make the process very simplified so that we can help families access this particular research. So lastly, I just want to mention an exciting announcement. So the mini study, as I said, has been going on for about 10 years or so, but we have this new initiative. We have seen patients with POLG in the past, but we have this new initiative actually, which is we recently received a very positive score for our U01 grant that we submitted to look at POLG disease specifically. And so the name of the grant is IMPRINT, which stands for Immune Mechanisms in POLG-related mitochondrial disease, inflammatory networks, and therapeutic targets. And the idea is that we are investigating this inflammation that we see in mitochondrial disease, including POLG disease, whether it is a contributor to disease pathology and actually may be a therapeutic target in the future. And so this is a study that is actually across NIH as well as outside of NIH. So this was initiated by Philip West, who's on the call today, who's from the Jackson Lab, who's an expert in inflammation as well as mitochondrial disease. Helen Su, who is an expert in inborn errors of immunity and immune dysfunction, as well as myself. And we are looking essentially for 20 families to visit us at the NIH Clinical Center. And as I mentioned, we provide all of the various aspects to help with travel and everything. Although we can see more than 20, but 20 is what we set in the grant. We want to see as many families as possible, actually. And the idea is to understand how inflammation and immune dysfunction may be possibly a driver of disease pathology in POLG disease, and whether we can actually intervene in this process in the future. And with that, I would be happy to take any questions in the chat, and I will respond to them.
Thank you, Peter. Amazing. Just incredible that you, Phil, and your team are doing this type of work. So 20 families. Any of you out there, please consider. Obviously, it'd be easy if you're on the East Coast, but I think, Peter, if I understand, you have travel funds available for
We do.
flights,
So we provide
domestic,
yes,
international,
domestic and international. We provide
and
travel
international.
clinical center. Yes. We provide food and housing and you know, all those things that just to make sure that families don't have to worry about those particular aspects of coming to us, because obviously travel in itself is a challenge to begin with, and these are removing some of the barriers.
Incredible. Okay, thank you. And please feel free to contact Peter and his team, questions in there, and always huge praise to Phil West and his amazing research and what's going on up at Jackson. I'm going to try to move on because I know we got 3 more talks of real importance. I'd like to invite Michio Harano from Columbia University to speak next. If I could get Michio to unmute, and I don't know if he wanted to show slides or not, but he's going to tell us some really important insights of a very hot topic in the POLG world. So please, Michio, take us away.
Okay, thanks. I hope you can hear me and see my slides. Can you hear me?
We can see the slides. We can hear you great.
Okay. Is it in slide mode for you? Okay. Okay. As long as you can see the slides, this is going to be a whirlwind journey here for 25 years of research in 10 minutes. So I'm going to buckle up. Okay, here we go. So I want to start with this diagram here, which just shows you some people call this the virtuous cycle. We start with patients, and I think all of us who are researchers here and clinicians are participating in this, identifying patients, getting to their genetic defects, understanding that how those mutations cause disease by studying cell and animal models, as we heard eloquently earlier, and developing therapies in the lab that we hope to take back to patients. So this is a cycle that we're all striving to achieve, and I think we've accomplished this at least in one round with TK2. TK2 stands for thymidine kinase, and deficiency of that was first described back in 2001 by an Israeli group led by Ann Sada, and they described 4 families with a very severe muscle disease. So these are young children, infants in most cases, who developed weakness that affected not only their skeletal muscles, but their respiratory muscles, and they progressed very rapidly, and they died of weakness of the respiratory muscles. So they died of respiratory failure. And this TK2, it's encoded by the nuclear DNA, and it's a recessive disease, meaning the parents are carriers, and the children unfortunately inherited 2 pathogenic variants, 1 from each parent. And the protein is here in the mitochondria. It phosphorylates these 2 nucleosides and makes the building blocks to make mitochondrial DNA. So this is very much related to POLG gamma, because these building blocks are what feed POLG gamma to make mitochondrial DNA. So you could use the analogy, they're just making the bricks that the POLG gamma, which is like a contractor, uses to build.
I don't give a shit. They're eventually going to figure it out.
We got someone else here talking. Okay, thank you. So anyway, yeah, so you could use this analogy of making the building blocks like bricks to make the house. In this case, it's the POLG gamma, it's the mitochondrial DNA, which is constructed by POLG gamma. So that's how it works. This is this is out of order. Let me just come back to that. So we and others identified other patients with TK2 deficiency, and it turns out to be like Paul gamma, diverse disease, not just affecting young children, but also there are some with childhood onset and some with even teenage, adolescent, or even adult onset disease. So the spectrum of disease varies. It's mainly a muscle disease, but it can also affect the brain, and especially the very early onset cases. The children often have seizures as well, in about 30% of the cases. And so we were able to characterize this disease in this paper that was led by Katerina Caron. And we also, very importantly, tried to go back and look at the course of the illness retrospectively. So taking a backwards look at the patients and their histories. And we saw that those with the very early onset had early, very rapid progression and early mortality, that is early death, unfortunately, of this disease, whereas those with the later onset, as shown in the red line here, had a slower progression and lived longer. So this was very important to understand the nature of the disease, the natural course of the disease, and that's exactly what the PIONEER study is doing, but in a better way, in a prospective way. So but retrospective and prospective natural history data are very important to understand the disease and to plan trials. So we were then fortunate to have a donation from a family to make a mouse model, and we made what's called a knock-in mouse model that had a mutation very similar to the human mutation, and it was very similar to the infantile onset disease in humans. With early onset, these mice stopped gaining weight at age 10 days, plateaued here, and then they died in the second and third weeks of life. So it's a very severe disease. And with that, we were able to show that there were imbalances of the nucleotide pool, the building blocks to make mitochondrial DNA, as shown here. So we decided to, since we had this model, to try a therapy, this nucleoside therapy, to give the substrates for TK2, which is mainly blocked here, but it goes through this other cytosolic pathway. So we're giving a molecular bypass therapy to make the building blocks necessary to synthesize mitochondrial DNA. So that was the idea. And Carlos Lopez here in the bottom right corner was the postdoc who showed that it was effective. It delayed the onset, as we heard earlier, with the G therapy, but in this case, it's a pharmacological therapy. And with that, in a dose-dependent fashion, it doubled or tripled the lifespan of the mice. So it was great. It delayed the onset, slowed the progression, and allowed the mice to live longer. So that was all done in our lab. So then, sorry, we're jumping ahead here. So while we were working with the mice, patients were coming to us, including this young child who had this unfortunate disease with the early onset. He was normal in the first year of life, but when he was about a year old, his grandmother noticed he had trouble holding up his head, and he quickly, in 3 months, lost the ability to sit and to cruise, that is, walking around holding onto furniture. And he was admitted to a local hospital. His CK was elevated, a sign of his muscle damage, and his lactate was elevated, sign of mitochondrial dysfunction. And he had a muscle biopsies that showed mitochondrial DNA depletion. And genetic testing showed he had these two pathogenic variants in TK2. So he came to us. We talked to the family, and he was unfortunately quite weak. He was about 17 months when he came to us. You could see how weak he is. He's a floppy child. And he was like a ragdoll. He couldn't lift his arms and legs off the table. So because the family was desperate and because we had evidence from a mouse model that this therapy could work, we actually started with the monophosphate, a slightly different variant of these building blocks. And then we switched him over to the nucleoside therapy. He started the therapy back in 2012, and I'm pleased to tell you he's still alive and he's doing well. Unfortunately, the day after we saw him, he was sent to the ICU, intubated, put on a feeding tube, but now he's able to get around in a wheelchair. So he has not recovered the ability to walk, but he has recovered the ability to breathe independently during the day, and he's going to school, and his parents were very proud. He won a science award recently. So we were were flooded with requests to try this therapy. We enrolled about 23 patients in our program and expanded access. And in Spain, they enrolled a similar number. But at that point, the FDA told us to stop enrolling patients under compassionate use and get patients into a real trial where you collect data under industry standards so that we could file that information to get the therapy potentially approved. So we were fortunate that we had an industry partner that was interested and took over and transferred these patients into a phase 2 trial. This is a complicated slide, but basically it shows you that we had data from untreated patients, both from the literature and then subsequently collected by the company UCB. And we also had patients who were treated early on a compassionate use basis, but then in a phase 2 trial, and then subsequently other patients who were identified too late for the trial, they were also given compassionate use compassionate use therapy. And so we were able to collect data and compare historical controls to these patients who were mainly treated in the clinical trial as well as compassionate use. And what the company did was they compared these historical controls in terms of survival. And these are the patients who were matched, so by age at onset, and treated untreated for a period of number of years. The blue line here shows the treated patients, and you can see there's a dramatic reduction of mortality. That is, they survive longer, 92 to 94% reduction in death with the treatment. So it really had a profound effect on the survival of these patients. And also it had a dramatic effect on the motor milestones, the ability to hold the head up, sit, walk, run, and so on. And before treatment, you can see here in the gray bars that more than 80% of the patients lost one or more of these critical motor milestones. After the initiation of treatment, some patients continue to lose some motor milestones here in the blue bars, but after initiation of treatment, only 2 patients regained motor milestones by the historical controls, but over 75% of these patients who were treated regained one or more milestones. So not only were they surviving longer, they were gaining motor function. So all of that was great and led to approvals of the FDA back in November and the European Medicines Agency for this treatment. This was the European agency approved this in January of this year. So this is fairly recent approvals, and we're very pleased to that it's reached now commercial drug status. Although we're now getting payers, hopefully insurance companies, to pay for it. Just to show you briefly here, hold on. This is an example of the young patients who start the therapy early. They do remarkably well because they have greater resilience and they're not as severely affected. This is a child that came to us from South Korea. He was about 10 months old when he started to show weakness, and here he is at 16 months, and you can see he's floppy like a rag doll, unfortunately. At this point, we were treating patients more aggressively than the initial patient in terms of the dosing and rapidity of escalation of the dose, and he started to move his arms better after only a month of therapy. After 3 months, you could see him, he actually looks more cheerful when you see his face. And he's able to play with this toy, and he's able to hold this object against gravity. He's not able to pull himself up to sit, but when he's put in a seated position, he's now able to sit upright and hold his head up. So here he is improving. And then this is him after 6 months. He's able to regain the ability to walk with a little bit of assistance here. And you can see that he's able to when propped up and ready to go, he could take a couple of wobbly steps to his mom. And after 10 months of therapy, you can see the same child walking around with his arms behind his back nonchalantly. He's not doing it because he I think he just wanted to show off at any rate. So just I think I wanted to talked about this because, well, Brian asked me, but I think there are important lessons that can be shared with across the spectrum for patients and families. Yeah, obviously, genetic testing is a starting point for diagnosis and development of therapy, and laboratory data, as we've heard repeatedly, is critical in order to understand the mechanism of the disease and develop rational therapies. Natural history data, whether it's retrospective or prospective, is critical to plan the clinical trials and to understand the disease. And teamwork, obviously, it's required many people across the laboratory, clinical work, industry, and patient advocacy groups. So it's really been a remarkable journey over the last, well, in my lab, 25 years since the disease was first identified. And I thank you for your attention.
Amazing, Michio. Thank you so much. And Michio, if you don't mind, if you're able to put your contact information, I know there's a lot of questions of this theme and what is applicable to POLG and what may not be, and it's where are the research gaps that we need to fill in. And I know there's some activities that are going to be very pertinent to this topic to be very active. For those of you that are keeping time check, it's clear we're going to go over. And I apologize about that, but I think for our next two talks, we just have to make sure they get their airtime. So with that, can I please invite Anthony Abruscato of Mighty Therapeutics, formerly known as Stealth Therapeutics, to jump in?
Yeah, thank you, Brian. Can everyone see my screen as I pull up my slides?
We sure can.
And I will do my best to be efficient on time. So good afternoon, everyone. I'm really happy to be with you all today, and thanks, Brian, for the invitation to speak. I'm Anthony Abruscato from Mighty Therapeutics. As Brian just mentioned, some of you may be more familiar with the name Stealth Biotherapeutics. We've recently changed our company name, but we're still the same team with the same mission. So at Mighty Therapeutics, we're really proud to have been working in the mitochondrial medicine space for so long. Our company formed back in 2006 around the promise of the drug elimipritide. And I'd like to share with you all some of our experience studying mitochondrial diseases, the lessons we've learned over the years, and how we're moving forward to tackle POLG disorders specifically. So elimipritide is a small peptide that has a very unique target. It's a phospholipid called cardiolipin, and this is exclusively found in the inner membrane of the mitochondria. So by binding to cardiolipin and stabilizing the inner membrane, mitochondria can function more efficiently and produce energy more effectively. Elimipritide is a very well-studied drug. It's been in development for a long time. We've enrolled nearly 2,000 participants across all of our clinical development programs, and it has a very well-characterized safety profile. And last September, elimipritide was approved for a very rare disease called Barth syndrome under what is called the accelerated approval pathway. So we have a variety of ongoing programs that we're studying either with elimipritide or one of our pipeline of compounds. I'll focus your attention to one of our longest standing programs in primary mitochondrial disorders. So in this clinical development program, we've conducted multiple clinical trials enrolling over 300 participants and spanning over a decade. So these two studies at the bottom were our largest interventional trials that were really intended to show the efficacy of elimipritide. So despite encouraging data from these trials, not a single one of them met their primary endpoint. As the saying goes, hindsight is 20/20. So when we look back at all of our trial experience, the signs clearly point us to POLG disorders. In our earlier trials, we enrolled an incredibly broad range of different genetic mutations and syndromes, all with a common feature of muscle weakness. We followed those data where the data were the strongest, and that led us to the new POWER trial, where we refined the population to a smaller grouping of different genetic diseases that all have a more similar function. Then last year, when the new POWER trial concluded, it became clear that our strongest signal of a positive treatment effect was coming from those participants with POLG disorders. And that's going to be our focus for a future trial. So why POLG with elimipritide? So I'm going to show a very quick video that really illustrates how the POLG enzyme is working within the mitochondria. So I'll just let this play for a few seconds. So you can see mitochondrial DNA replications really occurring right at the inner membrane of the mitochondria. And when the POLG enzyme doesn't work properly, you get less mitochondrial DNA, you get damage to the mitochondrial membrane, and you get severe deficiency in the electron transport chain, which leads to impaired energy production. So studies in POLG cells have shown that elimipritide protects the membrane, improves energy production, and even improves the amount of mitochondrial DNA that's produced. So when we look back at those participants with POLG disorders from our New Power trial, every outcome that we studied really suggests a favorable treatment effect with elimipritide across different types of exercise tests and a variety of questionnaires that are assessing symptoms of the disease. Any single one of these endpoints can be noisy or, not fully conclusive, but when independent measures are all telling the same story, it increases our confidence that we're seeing a real and meaningful treatment effect. So with a new study that's designed specifically for POLG disorders instead of a much broader population, we hope that we can confirm this effect with even more certainty. So where are we now? We've begun to discuss what a new trial in POLG disorders could look like with regulators. We have more meetings planned for later this year. We're looking forward to socializing our plans with the POLG community directly to gather critical feedback to make this trial successful. And we hope to initiate this study globally next year. So as we think about what a new trial looks like, I'd like to share some insights as to what we've learned over the years, why designing trials is so challenging, and I know we've heard from others on this topic as well, and really focus on what we can do to overcome those challenges. So we've seen many trials in mitochondrial diseases beyond just ours not meet their primary endpoints. And while these certainly were not the outcomes I think everyone was hoping for, it's difficult to suggest that any of these studies were failures. We've just learned so much about how to tackle mitochondrial diseases. And it's become, I think, somewhat clear that combining different mutations and syndromes, even if a drug should theoretically work well in all of them, just may not be a viable approach. These diseases are just incredibly complex and variable, and it's very hard to tackle them all at the same time. But recently, there have been 3 drugs approved for mitochondrial diseases. You just heard of one of them from Dr. Hirano. We have elimipritide for Barth syndrome, and we also have Skyclarys for Friedreich's ataxia. So I'm really encouraged that the field is moving in the right direction. So one of the biggest lessons we've learned is that designing a successful clinical trial is much more complicated than simply having a promising drug. Every study is a balancing act. You need the right patients, the right endpoints, the right duration of treatment, a feasible design for the participants and the investigators, statistical rigor, regulatory alignment, the list can go on. And what we found is sometimes these can actually be competing forces. So in rare diseases, all of these factors may overlap only in a very, very small space, and finding that sweet spot can be really challenging. So how do we fight back against these challenges? I'm just going to share a few strategies that we've implemented in our programs that I think have led to success. And I'll start here in the bottom right. And we've heard a lot about this, probably from half the talks today, about the value of registries and natural history studies. And these are just so critical in mitochondrial diseases to help us make informed decisions about how to design a clinical trial. And that's why, I think the Pioneer study is so brilliant and will really pay dividends for this community. sophisticated statistical analyses. As I was mentioning, any individual endpoint may really not be all that sensitive or perfectly tailored to a specific disease. So we've designed trials where we've actually generated composite endpoints where we look across multiple endpoints, and this may allow us to detect meaningful benefits where any one single endpoint might miss. External controls, we've heard about this today as well. This is really where you try to leverage existing data rather than just looking to your, a single trial in isolation. And these approaches are not easy to do, but if they're done correctly, they can really greatly help with the interpretation of clinical trial data. Novel endpoints. So sometimes the problem isn't the therapy, it's that we're measuring the wrong thing. So in some of our development programs, we've actually worked closely with patients to develop new outcome measures that really better capture what living with mitochondrial disease actually feels like. And of course, the patient voice, and again, something we've heard a lot about today, and really the value of initiatives like the patient-focused drug development meeting. We need strong partnerships between all of the stakeholders to make this work. It's essential. It's non-negotiable, and I don't think anyone could be successful without these partnerships. And the last thing I'll mention is, persistence and perhaps a little bit of stubbornness as well. We've learned that drug development is almost never a straight line, particularly in mitochondrial diseases. So we really have to be relentless and never give up. And with that, I'll turn it back over to Brian. Thank you again for the opportunity today.
Thank you so much, Anthony. If you could put down any contact information for Mighty, I'm sure there are people that would love to reach out to you. I see a number of questions popping in. All right. I do want to make sure I mention at the end of this summit, I know we're going to go over time. I apologize about that, but Heather Schichtel has offered to do a support debrief session after the summit where we'll stop the recording. Any of us who are not POLG families will drop out. So feel free to stay on if you're able to. And with that, I'd like to invite Ash Dogar of Pretzel Therapeutics to jump in. And I have your slides, I believe, available, Ash, if you want to.
That'd be great. I can
introduce
start
yourself. Yeah, please go for
Brian.
it.
So yeah, thanks, Brian, and the entire POLG Foundation team for inviting me to speak today. I've known Brian for a while, and he's been a phenomenal partner with Pretzel and clearly just across the community. So I'm really grateful for that. As he said, I'm the Chief Development Officer at Pretzel Therapeutics, and I'm so honored to share the time with these prolific researchers, clinicians, seeing the cutting edge work for the first time presented here, which is very cool. And also my industry colleagues presenting key program updates. Exciting time. Next slide, please. And you can go. These are forward-looking statements. Next slide. I'll try not to rush, but I also am mindful of time. So this is our mission. Really, those who aren't familiar with Pretzel Therapeutics, we're a clinical stage biotech company based in the U.S. with a research group in Sweden. Our core mission, as you can see here, this is what drives us each and every day, and this is really important for us. Next slide, please. Thank you. So our clinical stage investigational medicine is PX578, and that's what I'll focus on over the next 10 minutes or so. It's a small molecule, brain penetrating POLG activator. I'll describe that in a minute. And it's taken orally, and we're currently in phase 1 in healthy volunteers. So I think you've heard a lot about POLG. One point that I'd make is polymerase gamma is the sole DNA polymerase responsible for mitochondrial replication repair. And I bring this up because that is how we work. PX578 is a direct POLG activator and addresses that underlying disease-causing biology of POLG disease. So it's designed to stop and hopefully reverse disease progression across the entire spectrum of the population. At the recent UMDF conference, Pretzel's Dr. Julie Jones presented a pretty comprehensive set of key non-clinical data that we've generated in patient cells and animals. We don't have time today to recap all of that great information. So just very briefly, what we've seen in the non-clinical disease models is that PX578 can increase mitochondrial DNA when it's depleted due to various genetic mutations, including mutations in the POLG gene. And in patient cells, we've shown that PX578 has actually increased oxidative phosphorylation, has increased energy production. And then in two different mouse models that have low mitochondrial DNA, we've shown an increase in mitochondrial DNA and that we can improve function and survival. So these experiments are just very critical to make sure that we have a solid understanding of how PX578 works and how it can provide benefit. Next slide, please. You can go to the next slide. Thank you. So you've heard a lot about Pioneer. I don't know that we need to spend a lot of time on this. Danielle presented it brilliantly. And basically, this helps us prepare for clinical trials in pediatric and adult patients with POLG disease and characterize biomarkers and clinical assessments and patient-reported outcomes that are important to families and to patients. If you go to the next slide, please. So just very, very briefly, I'll probably double click on why this is so critical to advancing PX578 and other investigational medicines for the treatment of POLG disease. And we partnered with POLG Foundation and UMDF on this. So this is really important because as Anthony just said as well, it really helps establish baseline disease progression. So these natural history studies help define how a rare disease evolves without an intervention. And so this enables researchers to distinguish true treatment effects from sort of the underlying disease progression and variability associated with that disease progression. It's really important because it identifies meaningful endpoints and supports that trial design and feasibility. You've heard this theme a few times. So by tracking symptoms and outcomes over time, these studies help determine which measures, for example, biomarkers and functional scales, are most relevant and sensitive for clinical trial use. And data on disease duration, progression rates, and sort of the heterogeneity in patients, that variability in patients, that informs the trial design, the inclusion criteria, exclusion criteria, sample size, statistical calculations, optimal study duration, and many other aspects. Importantly, we also can use this for external or historical controls. So in rare diseases or ultra-rare diseases where placebo groups may be impractical or unethical, natural history data sets can serve as comparator arms to assess the effect of investigational therapies. It's also important to help clarify patient subtypes. So understanding that genetic, the physical or phenotypic or demographic differences, that helps us identify subpopulations that may respond differently to treatments, and that supports that precision medicine approach that we're all trying to get at. It definitely accelerates regulatory engagement and potentially approval. Regulators often rely on robust natural history data, especially in ultra-rare and rare disorders, to help contextualize clinical trial results, and especially when studies are very small or even they're single arm or open label, and just the treatment arm. So they need some way to compare that treatment effect against. And most importantly, or very importantly, it improves patient-centered drug development. And this is a theme you've heard throughout the day. That is, natural history data, when done well, as in Pioneer, extracts what kind of clinical and quality of life outcomes are most meaningful to patients' lives. And this is really critical, not just for sponsors as we design trials, but for regulators that require this information, as do payers. And this goes into their evaluation of drug approval and reimbursement decisions. Next slide, please. So I'm just going to transition into some of the clinical development considerations and regulatory foundations in developing a novel agent that will hopefully provide context for our stepwise approach for developing PX578. And one thing I just want to mention is the urgency to move as fast as possible for all patients, especially the pediatric population, is front and center for me and everyone at Pretzel. So I just want to spend a few minutes to talk about some of those aspects that we have to think about and do to ultimately get our treatment to as many patients who need them as quickly as possible. So we know that the earlier age of onset, the greater the severity of POLG disease progression. And we understand the urgent need to treat, young children and adolescents. So we're committed to developing PX578 for all patients. So drug companies, just generally speaking, typically test investigational treatments in adults before studying them in children to meet regulatory requirements, ensure safety, and generate foundational data. There are exceptions to the rule, and you've heard of some of that even today. Regulatory authorities like the FDA generally require that initial safety data in adults is obtained before pediatric studies can begin. And this just reflects a long-standing framework designed to protect children as a more vulnerable population. So even in rare diseases, this stepwise approach helps to de-risk pediatric research, improve trial design, and ultimately increases the likelihood that all children, all adolescents, all adults will receive treatments that are both safe and effective. Next slide, please. So PX578 specifically is being tested in healthy adults. And there are steps that we have to take before we can safely test it in children, including evaluating in adults living with POLG disease. And so some of these considerations are listed here. So extensive animal safety studies are required, including in juvenile animals. So we and many other sponsors have to do both short-term and long-term studies in animals called toxicology studies or safety studies, studies that could take 6 months or 9 months in duration, and in more than one animal species to allow for chronic dosing because this will be a lifelong treatment. And a little more specifically, often juvenile or almost always juvenile animal toxicology studies are required to dose the very young ages. And that offers critical data on potential risks on all organ systems. Adult data is really critical, as I mentioned, to inform our approach to the pediatric population. Adults' fully developed bodies allow more predictable more predictable understanding of what the drug does in terms of how the body absorbs the drug, how it distributes it across the body, across tissues, across muscles, how it metabolizes the drug, how it breaks it down, and how it clears it from the body. So understanding this reduces sort of those risks that you may potentially find as you get into younger and younger patients. So adult safety and pharmacokinetic data, meaning like how the body goes through the system, biomarker data and efficacy data, that all guides pediatric dosing to ensure safety and inform how best to go into younger patients to ensure you have the right benefit risk profile. And so regulatory safety requirements often lead to the stepwise pediatric study approach. And especially for first-in-class agents, and I really want to emphasize PX578 is novel chemistry. It's a first-in-class. There's nothing like this kind of chemistry that's been out there. So that adds a level of scrutiny. So health authorities often require safety data in adults before allowing trials in children, unless there's very strong scientific reasons to justify otherwise. Adults also, provide a more acceptable starting point to understand how a therapy behaves in the body and to identify those potential side effects. So these data inform dosing and safety monitoring for pediatric trials. And I think as you heard from Dr. McGuire earlier, children are not simply small adults, right? They have a different physiology, different ways of interacting with drugs and experimental drugs and drugs that are approved. So it's really important to understand what the drug is doing in adults first. And because their bodies are still developing, those drugs can work differently and may carry unique risks. So the animal studies, looking at adults with POLG, all of that helps us be able to safely dose in younger and younger patients. And really, this is important because there's enhanced protections for children. There's heightened scrutiny and strict risk-benefit standards from ethics committees and institutional review boards that review what you want to do. And these are all designed to protect all patients, especially children. So studying treatments in adults helps reduce sort of the uncertainty and minimizes the risk of severe side effects before introducing them in children. So you're really trying to protect those pediatric patients later on. And, if we were to go into pediatric patients without any understanding of the benefit risk profile from animal studies or from looking at the drug in adult patients first, and there was an unfavorable outcome, it could significantly slow the entire program and potentially delay getting the treatment to anyone. And we want to be able to be, mindful of that and thoughtful of that. So while PX578 has been tested in healthy adults, we have to have information about how the drug acts in adults living with POLG disease so we understand its actions in the body and gain critical data on its benefits and risks. And again, this will help us determine how best to dose in younger patients and ensure that we can get the drug safely to them and that it can provide benefit. And I understand that, time is of the essence. There's a tremendous sense of urgency from the community, from us, and it's imparted to us by your voices very clearly. And we're trying to work as fast as we can to get through some of these steps that we have to get through to be able to bring this drug into patients. Next slide, please. And so this is just quickly where we are at right now. This year, we're completing our healthy volunteer study, and we're going to initiate our study in adults living with POLG, but also doing the work, the foundational work, so that we can understand the profile of the drug and get into younger and younger patients as quickly as possible. We've had a number of great engagements with regulatory authorities across the globe, including the FDA, to be able to explain what we're doing, why we're doing it. And so we're getting a great alignment with regulatory agencies in terms of how to best develop the drug for all ages. So with that, I'll stop and thank you again. Sorry that it was a bit rushed, but I'm mindful that people are dropping off and trying to get that information out to you right now. So thank you so much. Thank you, Brian.
Thank you, Ash. Just incredible. So grateful for you to take the time and share your insights and speaking on behalf of Pretzel, which we know you're working hard. So taking time to come and talk to us is time away from the work we really want you to be doing. So we get that. I'm going to kind of take an executive decision if it's okay with you, Ash. I have a slew of questions that various families have emailed me. Would it be okay with you and the families that I have you address these questions potentially in writing, and you can send them back to me or post it up on Pretzel's website? Would that be okay? I just want to be respectful of everyone's time, and it's the weekend. And would that be acceptable to you? And I hope no families are going to be grumpy with me if I do this. But I think it's very, very helpful what you just shared. So with
It's
that,
definitely, sorry, Brian. Yeah, absolutely fine with me. And also happy to engage with families as needed. So absolutely. Okay.
Okay. And the last thing I'd like to wrap up with is really a thank you to each and every one of you. You are not alone. We are global and we're together. We're on this mission. We got to solve POLG together. So I thank each and every one of you for calling in, for every speaker, for UCB, and congrats on the TK2 work, for Mighty Therapeutics, Pretzel Therapeutics. And I have to point out this lovely lady here, Paula Casanova. Paula, are you still on the call? Could you please say hi to everyone if you're here still? I hope you are. But here she is in this photo with Prince Frederick, who passed away last year, and she's been integral to the POLG Foundation and its launch and where we are today. Couldn't have done it without Paula, and this will be her last month with us. So we want to thank her. So if any of you know Paula, give her some hearts and love and clap some hands. But other than that, thank you all, and I look forward to seeing all this amazing research move forward as fast as possible. And let's do this together. Okay? I wish you all a great weekend. Thank you again to all the amazing speakers. That was some incredible science, and I'm pretty dazzled. So I hope you guys are going into the weekend with some optimism that POLG is not being ignored. Thank you, everyone.